Well, thank you for joining us tonight. Tonight, we're going to be talking about this research that was community-led here in the Northwest. But before we get into that, I just wanted to go through a brief outline. So I have a few announcements that I want to make sure everyone knows about. And then I have a gene therapy trial that's going to be conducted within the ACTG. And I wanted to review that with you all. And then we'll get to our main presentation. And so we're going to skip that just because I want to skip that. The first announcement I have, if you're not aware of it, Pier Seattle, which is on Capitol Hill on Bellevue Avenue between Pike and Pine. And they're having an open house Monday, February 6th, starting at 5 p.m., going to 9 p.m. And they will be giving out goodie bags. They have swag bags that will be able for people to take home while they have those supplies that last. So if you're living in the Seattle area, you should check out Pier Seattle. I think it's a great group. This is important for everyone, whether you are in Seattle or anywhere in the world, you can join this. This is going to be an event put on by the Reunion Project. It's a virtual event, and it's going to be on February 16th, 11 a.m. Pacific time, 2 p.m. East Coast time. And it's going to be participating in clinical research for long-term survivors of HIV. So it's going to talk about specific issues that long-term survivors face. Now you see that QR code to the right. That's an easy way. If you have a smartphone by your side right now, you can scan that QR code and it'll take you to the registration page for the clinical research event that the Reunion Project is putting on. But I think it's going to be a great event. And if you have not heard of it, they will be having a panel. They'll be discussing the issues of around long-term survivors of HIV. Sometimes those are age related, but they're also people who are vertical cases. In other words, they may just be 30, but still have issues that are specific to them in terms of being long-term survivors who were born with HIV. So that's the first event. That's a national event for you all. And this is the next event that I wanted to plug. On Saturday, February 18th, here in Seattle, as well as virtually, we'll be having the 2023 Community HIV Cure Research Workshop. So it'll be from 8 a.m. to 4 p.m. It'll be at the Crown Plaza, downtown Seattle, and there will also be a virtual link. So if you are not coming to Croy this year, you can still join us on that Saturday from wherever you are, provided that you have an internet connection. So again, you'll see the QR code to the right if you are interested in registering for that. Whether you want to be virtual or you want to be in person, we would like to know. And so we're trying to ask people to virtually join us. So if you want to take a snapshot of that with your cameras, if you have a smartphone, that'll be the way. I can also send this out via email. I may have already sent it to a lot of you that way, but it should be an interesting meeting. We're still putting the agenda together, but we're going to have a specific session on long-term survivors and HIV cure research, specifically issues around ATIs and things like that that may affect the participation from long-term survivors. Other issues as well. We're also going to go over the clinical landscape of cure research that's going on, and we're going to be looking at special populations as well, the key populations and how cure research is affecting them. So to go into the study that I wanted to review, the ACTG has a study. However, this study is going to be a gene therapy study. It's going to be at one site, however, and that is in Pennsylvania, specifically in Philadelphia at the University of Penn. It's a small study, and it's going to involve 12 people. So 12 people total, and they're going to be broken up into two groups. So there'll be 6 in each group. And what they're doing is testing a new way to possibly treat HIV, which is taking a person's T cells and modifying them. So they collect the T cells, then they modify them in the lab so that the cells can find and target HIV infected cells, and then they're placed back into the body and set to do the work. Now, the official term for these are HIV-resistant, HIV-specific dual CAR T cells. We could just call them CAR T cells for short. The dual part of that just means that they have 2 different mechanisms through which they're trying to do their work. So this is the step 1. People will go through this process where they have their T cells removed, modified in a lab, and then they will be given back to them through an infusion. The step 2, at 4 weeks and then at 12 weeks, half of the participants will receive a booster vaccine and the other half will receive a placebo. And it's a DNA vaccine. And the question that they're looking to answer is, will giving them a booster vaccine improve the T cell activity? Will it make these CAR T cells work better? And so these vaccines are going to be delivered through electroporation, which is basically giving very short electrical signals or pulses to the muscle to get the cells to open up so you can get the DNA vaccine into the cells. And so that is what's going to happen during step 2 of this trial. And then the third step is an analytic treatment interruption, which is for 16 weeks. And that will start 4 weeks after the second injection. So that was at 12 weeks. So this is going to be 16 weeks, and it lasts for 16 weeks. And so if people have a viral load over 100,000 copies and they have that for at least 3 weeks, participants will be expected to restart ART. Now, there'll also be a step 4 for some participants who may qualify for it, and that's to continue the ATI for up to 8 more weeks if their viral load stays low, which is described as under 1,000 copies per milliliter of blood. Step 5 is when participants are ready to restart, they will enter step 5 for monthly study visits until their viral load is undetectable again. And then they will have visits every 6 months for the remainder of the study. And the study is scheduled to go for 5 years. So this study is only being done at the University of Penn, and it's a way for the ACTG to try to branch out and do small pilot studies that might be only specific to one area. And Penn is well suited to do these gene therapy trials, and so that's why it's being done just in Penn. But I don't know if just hearing what I've shared with you all, if you have any comments or questions or anything that you want to know about the study. This is a schema of how the study will work. I verbally described it to you, and now you can kind of see it pictured. So step 1 is when they're going to be having their cells modified, and then day 0 there is when they will be given those infusions of those cells. And then they have the vaccine at week 4, week 12 during step 2. Step 3 is the treatment interruption. Step 4 is an extended treatment interruption. And then step 5 is when they restart ART. And then they have the secondary follow-up, which is supposed to go on every 6 months for the next 5 years. So you can kind of visually see how it's going to go that way. I just wanted to share that this study is being done with the ACTG. It's one of those things that I was trying to be a little sensitive. A lot of people in our local Seattle community are feeling a little bit left behind by cure research and by the sort of strictures around the study that keep them from ever being able to participate. And so knowing that this study was never going to be coming to Seattle or anywhere really else besides Penn, I figured I would want to review this with you, but not really spend too much time focusing on it. But I didn't know if anyone had any questions or comments they wanted to make having being presented this information. I know I went through it kind of quickly.
I guess the question I have is, what are they expecting the vaccine to do on top of the CAR T cells? And, you know, kind of given that so far we haven't had much luck with vaccines, you know, is there data to indicate that it's gonna make much of a difference?
Yeah. So they do in the study protocol draft that they shared with us. They say that this DNA vaccine is about getting the T cells active. So I think it's the combination of the modified cells and then giving them this boost that way, but they're not sure. Mm-hmm. That's why they're doing this experiment. They don't know if it's going to have an effect or not. Mm-hmm. But there was some validation that was in the protocol.
And they have ways of measuring whether or not just the CAR T cells themselves made a difference, because it looks like they're not being compared to no CAR T cells.
Right, that's true. So the randomization in this case is to the vaccine or the placebo, not to the CAR T cells. Everyone is getting the CAR T cells. So I would assume that, but that's a good question to ask. Thank you for sharing that.
My guess is that there's a pre-post analysis within each individual person. So they're probably comparing the person's cells when at the time of screening or when they remove, right? Because they're removing their own cells. So they're probably comparing a variety of things for someone's viral load and CD4 count and things like that. But I haven't seen, I would have to actually pull up the actual measurements from the study. I can do that if people are interested in it. But that's my guess, is that this is that there's a pre-post analysis within each individual person.
That makes sense.
I very much would like to hear more details about this study. If you're able to communicate through email with us or with me, that will be fine.
Sure.
I have a
I might pop something into the chat. I'll go onto the study website and pull the protocol and look and see how they're planning to compare the
I think
outcomes.
Since it is so far away from Seattle, Washington, I will just directly learn from the website and through the study itself.
Were there any other comments, questions anyone wanted to make? And I can share with you all right now. Let me get this chosen.
Michael, and do you know if they're also going to monitor for detecting the dual CAR T cells over time in the participants?
Say, what was your question again, Mark? I didn't get that.
That they're actually going to monitor for the survival of the dual CAR T cells, because I understand there's ways that they can look and distinguish them from other T cells. So are they going to monitor those as well?
Yeah, absolutely.
Yes. And I think that's, so they say that this is going to be, the follow-up is for 5 years. So that's one of the things every 6 months that they're going to be doing the blood draws to look at that sort of stuff. You know, so at least for the 5 years of this study, they're going to be looking at the people. You know, there are, I don't know if I should say rules, there are guidances that suggest that people should be followed for 15 years after a gene therapy trial. I don't know if that's going to change, but you know, this study is at least going on for 5 years, and most of the 5 years is going to be following up. You know, they only get that one infusion way back at the beginning of the study, and then the rest of it's really just sort of seeing what effects have happened and how long these cells will last. But yes, they're going to track that.
What is the risk factor with this? It strikes me that they're only doing one study because it must be a high risk factor.
Well, it depends on what you see as the risk factor. So the dual CAR T cells are currently being used in other people. So they know that the duo CAR T cells themselves are safe. So there's no fear about that. Of course, the ATI is the treatment interruption is the part that we worry about the most for not only a person's health, but also for the health of their partners around them that they may have, especially if they're sexual partners. And so that would be probably the riskier part, I would say, in terms of the fact that who knows what could happen, you know, but we don't have any concerns about the gene therapy product. We know that that's being given to people and that's safe. So that part is okay. It's the treatment interruption, I think, that is the most dangerous. And then you could also say the vaccine, while it's not dangerous, I know people who've been electroporated and it's not a pleasant experience. You know, it's not that it's not safe, it's just not very pleasant for people. The muscle contractions that happen with that procedure makes it hard on people.
I think this is also a first in human study. So this would be a standard size for the first time of giving a particular product to people.
Yeah.
So there is the possibility that it'll expand if it's showing promise.
That's the question that I've asked. I don't know if it will expand. You mean the study, if the study will open up to other people?
I think unlikely that this study, because this study is designed as such with this fixed sample size. I think generally it would move to a larger phase study. I think the other probably big rationale in having it be so small is that yellow portion that says cell manufacturing.
Mm-hmm.
I don't know what the cost is of removing someone's T cells and doing gene therapy. Maybe actually one of our guests, either Jack or Jared, know that what the cost would be of doing the cell manufacturing, but it is quite huge. And so I imagine that they want to start with a small study to see if it's safe and if the cells persist and are HIV specific in a person before they made a larger study. I don't know, Jared or Jack, do you know anything about cost of just doing the cell manufacturing?
I can't give any numbers specifically because I don't work quite in that field, but I agree with you that it's a very expensive and very arduous process to manufacture those cells.
Right. And that's one of the ways that a group that I volunteer with called the Global Gene Therapy Initiative is looking at changing that part of the whole process to make it less expensive. In other words, the manufacturing of these gene modified cells needs special labs to do it, but through Jenna Dare and her work with the Prodigy machines, making basically, if you remember back in the day when we used to talk about gene therapy in a box, and trying to make the manufacturing of these cells local. So like, in other words, right now, Penn can make these, and I think it's involving, I don't know if it's involving Case Western. I might be confusing this with the other dual CAR T cell study that I know of. But that would be a problem to expand this study. Like, in other words, if this shows promise in terms of a therapy that may even be curative or lead to a cure, the problem is, can we expand this further? The ACTG, even domestically, would have problems having this study expand that way, let alone bringing it to the international group that the ACTG is made up of. Like doing these in limited resource settings, such as Thailand or Africa, anywhere in Africa, Uganda, India, this study would be not feasible.
Well, I was thinking, as you were talking, that this sounds just like Jenna Dare's Cure in a Box. I am excited to hear that something is finally moving, because it seems like so long ago when we heard Jenna Dare talk about her theory. So
is she still doing research on it, or is that topic still an active topic?
Go ahead.
I can speak to this. Yeah. So there are a few members of the lab still working on this, and it's more about settings. Like, for example, we have a fellow from Uganda at the moment trained on this technology. We're planning on taking it back to their site over there. But other than that, our work is more in stem cells than T cells, as is the case here.
I see Kareen's hand is up.
Yeah, Kareen, I was going to say, I saw your hand was up.
Yeah, so Michael, can you clarify? You mentioned this is a 5-year study, but I thought the period of follow-up for people who received cell and gene therapy product was 15 years. So would that then fall back on the site to make sure this happens?
I don't think that has been worked out at all. It's a question that I've asked to the study team myself. It's not clear. So this has that 5-year built in. So does that 15 years start after these 5 years? Or are they building in these 5 years as partially that monitoring? In other words, it sounds like the secondary follow-up, the step 6 part, which is going to go on and fill the 5 years, it sounds like that's what you would be expecting in terms of the monitoring of these cells in people's. But yes, again, this is about safety, as you can see if you looked in the chat there. So I don't know the answer to your question, Kareen.
Thank you.
Sure.
It just popped into the chat a few things about the secondary objectives. So the primary objectives of the study is safety of both the vaccine and the CAR T cells. So separately, the vaccine and the T cells. And then the secondary outcomes are comparing the individual's parameters to themselves. So they're looking at HIV specific cells before they get the infusion, after they get vaccinated and after the ATI, they're looking at different reservoir measurements, intact proviral DNA, things like that, before getting the infusion and after being resuppressed on ART after their ATI, and various different levels of virus and measurements of CD4 counts and all of these things. So it looks like they're really comparing people within each arm to themselves, from the like yellow period to the green period to the purple period to the blue period, and then all the way through the end, and then also comparing between the groups about whether or not getting that extra vaccine makes a difference in any of that.
Does it address Corinne's question at all about the duration of the follow-up?
The monitoring is I'll look through it again, but I think it is it looks like a 5-year study. What were you saying, Michael, is that the mandate is that all that CAR T therapy or gene therapy are supposed to last for 5 years?
No. So
supposed to last for 15?
Yeah. The guidance is supposed to be that there's supposed to be follow-up for 15 years, but we've already seen how this is not happening. So for instance, the very famous Sangamo zinc finger studies, there were results that happened that haven't been reported because they were dropped, because Sangamo decided to drop HIV from their repertoire, so to speak. And so those people went unfollowed. And yet we were hearing anecdotally from participants in that study how they had a T cell boost and that they were doing healthy. And there was even, maybe not in terms of a curative therapy, but it was definitely helping people who were immunologic non-responders. And so there was a lot of interest in the community around that, but it seemed like people were just dropped. You know what I mean? There was no follow-up around that. And so that's why that comes up as a concern for a lot of us to think about.
Yeah. Let me do some digging, because okay, so it looks as though this study that has these different components to it, so it's a little bit hard to read, because the timing of each of the pieces is sort of with respect to the prior piece. You can see that it re-zeros at each step, but it seems as though step 6, secondary follow-up, starts at 12 months and is every 6 months up to month 60 post-infusion.
Right.
So from the beginning of receiving the gene therapy. So I don't know. I mean, there isn't really a way to follow people off of a study other than to possibly receive safety information if somebody calls in. I wonder if, this protocol is pretty clearly set at 5 years right now. I wonder if the plan is to get this out the door and then potentially they only have funding for 5 years, and then to amend it for a longer follow-up or something like that. I'm not sure. We'd have to ask.
I think a lot of long-term publicly funded studies just get 5 years of funding at a time, and then you just add on another 5-year extension and a 5-year extension. But I don't know if that's what their plan is.
Yeah,
Michael,
and I think good point from Richard too about it's easy to follow people up for safety, but to do those detailed assays to look at does the product actually work and verify some of these things that participants were saying about their immunologic status and their viral control. I think it sounds like Sangamo didn't do that. They did follow people for safety for longer, but yeah, I think Laurie, you're right. My guess, and that's my guess, is that it will be a different funding period that will end up tacking on additional follow.
And I also,
Michael, whose recommendation is the 15 years?
I don't know. I don't know exactly where that came from. Does anyone know? FDA.
I think FDA has some guidance. I will send it. The FDA is just posting guidance on cell and gene therapy. Like, there's no tomorrow. I'll send the link.
Okay, great. And also, just to point out in the chat, if you haven't seen it, Richard was saying that there has been eventually a long-term safety study for the participation in the Sangamo trials. And also, I did put a copy of the CAB draft of this protocol in the chat for those of you who are interested in looking at it further. You can get that downloaded from the chat right to your computer. So feel free to take a look at that. And Corrine just posted that that link from the FDA. Thank you for that, Corrine. So just two last questions that the ACTG likes us to ask. So do you think this study is going to answer an important question for the HIV community? How do you think? Do you think this is important?
Yes.
Yeah. And do you have any suggestions for changes or things that you had further questions about with the study?
Are they providing PrEP for partners if people have steady partners?
That's a good question. I don't think that the ACTG has a formal mechanism yet for that. And I know Corrine could probably help talk about that a little bit more because she's working with their working group that looks at that. But I don't think it's an official provision made by the study. Corrine, am I right with that?
I think they defer to the sites, but we're trying to work on more ACTG-wide guidance for partner protection. So Dr. Michael Peluso and myself are co-leading the partner protection working group as a volunteer effort right now. So right now, I think they're referring partners to PrEP, but it's very heterogeneous across sites. But I think Penn has a capability to do that.
Yeah, and that is what we're doing here. So for example, for 53, 57, where we're giving people BNABs, is that we are counseling people about partners and making sure that, if they have, for example, multiple partners that or people that they can't have a conversation with about PrEP, then potentially discouraging people from participating if they don't think they can sort of negotiate partner protection in some way about it. But then we've been sort of providing warm handoffs to Madison Clinic to help get partners on PrEP who are willing.
Mm-hmm. Also, I just wanted to point out that Richard put in the chat that he's uncomfortable with the viral loads being allowed to go up to 100,000 before there's any sort of and it's for 3 weeks that they could have that viral load before they go back for a restart criteria. How do other people feel about that?
Seems high.
It does seem high.
Wasn't there a group that talked about that and then talked about wanting to increase the viral load? And I don't know if it went as high as 100,000.
I don't know.
But I thought with the dual CAR T cells that they actually work better if there was virus because it would challenge them to function and expand. So I don't know if that would be a bad thing.
Right. Yeah. So there was concerns with the expansion of the CAR T cells and giving them something to respond to.
I just kind of worry a bit about that. There's going to be a lot of inflammation associated with that, and there are some older, studies of treatment interruptions where people had heart attacks, and they weren't really attributed to the interruption until after the SMART results became available. I mean, maybe it's excessive to think that might be a risk, but it just there seems to have been a sort of shift from very conservative ATI criteria back. It seems to be slipping sort of increasingly liberal, and I just worry about something bad happening.
Yeah.
I think these are valid concerns in terms of health and safety. I think the allowance for viral rebound really depends on if you're trying to conduct a viral set point study or a time to viral rebound study, and what you think that your intervention is going to achieve. If you think your intervention is going to prevent people from rebounding, then you can have a really stringent criteria of like, as soon as they've rebounded, put them back on ART. But for other studies, wanting to see, can you introduce control of HIV within that individual, then you kind of need to allow for them to undergo a viral rebound to see if they do achieve viral set point. And I do agree that I think these CAR T cell studies work best in the background of antigen for the immune system to see, which I assume is why they're including that vaccine as part of the study to really kind of boost the responses.
Well, thank you for sharing, everyone, what you did here. I wanted to move on to our main presentation. And so by way of transition, I just wanted to introduce you to Laurie. If you haven't met Laurie before, I think most people on this call have, but Laurie has been a longtime CAB member of this CAB and of the Defeat HIV CAB, and has also been involved in doing community-led research, qualitative research, and you'll see two examples here. One is that Facebook post from the research we did, it seems a million years ago now, where we called it "We're Scared and Brave." And if you remember that, we did that at Gay City. And then more recently in 2019, she presented at the Cell and Gene Therapy Conference that year. You want me to what? Results from a qualitative study that we did about the acceptability of gene therapy. And so I'm going to turn this presentation over to Laurie. Laurie, thank you for doing this.
All right, thank you. It's my pleasure. Thank you for having me. I just want to request, if you're not talking or asking a question, if folks would mute so we don't have a lot of background noise.
Sure.
So this was a study. We had wanted to call it "It Gives Me Pause" as with the preface, because that was kind of the overall response we got to a lot of the questions we asked, but the journal editors thought it might have been clickbaity, so we had to give it a more neutral title. So I want to start with thanking my co-researchers here, Hirsch and Michael and Jane and Karine. And Michael, you want to go to the next slide?
Sure.
So I have nothing to disclose. No one's given me any money. And then next slide. I want to start with a community summary slide where we kind of developed this as a best practice for scientific meetings. It makes a huge difference when people that are discussing sciencey science do this for us, which is to talk about what is it we were trying to find out, what did we learn, and you know, why this is important or exciting. Or so we know that what we wanted to do was find out how people who are providing care to people with HIV or with HIV feel about an HIV cure, how feasible they think it is, what do they know about HIV cure research, what do they know about cell and gene therapy, and their thoughts and feelings about that, and what might lead them to either encourage, discourage, or support participation in their patients participating in cure trials. And so really what we learned was that providers do understand the importance of a cure to their patients. They think a lot of the importance of that is really socio-emotional and related to stigma and other issues. But also there are a number of people, you know, for whom taking HIV meds consistently really is not an option. And overwhelmingly, they support autonomy and patient choice about making decisions about participating in research. And both factors related to the study, what it is, how it's being conducted, what are they looking at, as well as factors about their particular patients. How stable are they? How healthy are they? All of those things would contribute to whether or not they would be supportive of their patients participating in trials. And this is really important because what we learned during that "You Want Me to What" study, which grew out of the Defeat HIV CAB, when we asked people with HIV how they would feel about participating in a cell and gene therapy trial, many of them said they would ask their providers. They said, I would talk to my doctor first before deciding. So we know that clinicians are really important gatekeepers for trials. And so understanding what's important to them will help us design better trials, and trials that we're more likely to get more robust participation in. and we need participation in order to answer the research questions. So that's why this is important. Next. So our study next is, as I said, to learn how the community clinicians perceive HIV cure and cure research and what would lead them to encourage somebody or not. Next slide.
Yeah, and oh, go ahead.
Go ahead.
I just wanted to bring up what we meant by community clinician. How do we, what is the big separation between a community clinician and another type of clinician?
So what we needed to do in this study was differentiate between those who are conducting HIV cure research, because there are many people who are clinicians who are also HIV cure researchers, and those who are not actively involved in doing that research. So we were making, thank you for asking that. So we were making that distinction. So if I'm not involved in research, cure research, and I'm taking care of somebody with HIV, how do I think about this?
Right.
And how would I interact with patients who might want to participate in a trial? So that's really what we set out to learn in this study. So next. And again, this really grew out of that other study we did when people said, I would ask my doctor. And then the other piece of that is when we looked in the literature, you know, when people talk about stakeholders and doing stakeholder research, there was a lot about how people with HIV feel about cure and cure research, but there was very little about how people who are taking care of people with HIV view this. And so it seemed like a real place where we could make a meaningful contribution. Next. So what we did was we reached out to, we used what we call a convenience sample. So basically the four of us really sat down, five of us, and said, okay, who do we know who's providing HIV care in the area? And we made a list. And we reached out to about 70 or so people. In addition to the list we made up, everybody who agreed to be in the study that we interviewed, we also asked those people for names of people they thought we might want to contact. And then we reached out to them using the name of the person we had interviewed, hoping that that would get us a yes. And we did get some folks through that mechanism as well. So we wound up getting a total of 12 people, 11 of whom I was able to interview. I interviewed them on Zoom, and one person responded to the interview guide in writing. We had a few more people that had said they would do that, and then I think once they saw the questions, they just said, oh no, this is too much, and never did it. So we conducted the interviews on Zoom, we transcribed the results, and then what we did was we do what we call coding, where we organize them into different themes that came out from what people said. So that was the basic thing. Next slide. And this is who we talked to. There was a rather broad range of folks. We have people with all kinds of different levels of experience, from newer providers who are only providing care for, say, 3 years, to people who've been doing this for more than 25 years, people who are in community health centers and hospital clinics and private practice, different kinds of providers ranging, you know, we had infectious disease specialists, family medicine people, nurse practitioners, pharmacists. The providers are mostly white, which is what we see in terms of who's providing care. And it was fairly mixed in terms of gender. So we felt like we got a pretty representative sample of the folks who are providing care. Next. So what did we learn from them? So the first thing, next slide, is that most people had previously supported some kind of research participation by their patients. And by and large, people felt that, you know, patients had the right to make their own decisions about whether or not they wanted to participate in trials. Many of them felt like it was not their role to encourage participation in research. They really felt that being somebody's care provider is centering the patient, and promoting research may not be centering the patient necessarily. So they didn't feel like it was their role to encourage participation in research, but they were willing to support it. And some people acknowledge that they often didn't even know what kind of research studies were open to people. So I am going to apologize in advance. There are a lot of words on these slides. And the reason for that is we want to share with you what people actually said. So the slides are a bit wordy. So next slide. So some of the things that affected trial participation, again, many of them were patient factors. They wanted to make sure their patients were not being motivated by money, that they really understood what the study goals were. They were concerned about what the study goals were, what were the requirements and the logistics, what kind of burdens were going to be put on the patient, what were the potential risks, potential clinical benefits. There was a lot of concern around how it might impact the clinician-patient relationship, and I'll talk more about that. And very much people were concerned about the patient's clinical profile, their immunologic profile. And some actually remembered the early days when being in a trial was the best way to access whatever was, new and coming in HIV treatment, when there were very few options available. Next slide, please.
Bill has his hand raised. Bill, did you have a question?
Oh, I'm sorry, Bill, go ahead.
It really disturbs me that all the people that were interviewed were white.
There were 2 who were Asian, but that's who agreed to participate in the study. And I would say that the majority of HIV care providers in the region are white. If you look at who's prescribing, certainly not all, but a very large number.
Yeah, I understand that. I just wonder if that impacts the outcome in any way.
Mm-hmm.
But it was an observation that just jumped right out at me. So I felt like I had to say something.
Yeah, okay, well, thank you. Okay, so next slide.
This one is up.
Oh, okay. Yeah, so we asked them about their belief in the likelihood of cure discovery. A number of them felt like it was possible. Most people felt like an eliminating cure, where HIV is completely eliminated from the body, was a very high bar and a long way off. People felt, by and large, that durable art-free suppression, where you don't need to be on medication, but where the virus isn't active and isn't doing any damage, was much more likely. But there was definitely buy into the possibility of a cure. Next. So they did not feel like the cure was urgent, with there being much better medications now. We've got long actings that are now coming on board. There are higher barriers to resistance and fewer side effects. So they didn't feel like, oh my God, we need a cure today. But they did acknowledge the challenges that people still have. And everybody understood how important it is to patients, and they all had patients that had asked them about cure. Next, please. They really felt, by and large, most folks were kind of minimally knowledgeable about HIV cure research, and even less knowledgeable when it came to cell and gene therapy. People had heard about Timothy Ray Brown. Some people had heard about CRISPR and BNABs. There were a couple of people we talked to that were very knowledgeable. But by and large, they've got so much to keep up on, and treatment is continuing to evolve, that really, if they're going to a conference, if they're going to read an article, if they're going to do something, they're much more focused on things that they think will have an immediate impact on their practice rather than something that feels very down the road to them. Although the pace at which we developed the COVID vaccine actually had some people thinking, oh, well, maybe science can move a little faster, than I think it does. The other thing a few people mentioned is that the science is really very complex and hard to follow. Most community clinicians are not doing bench research, and so some of the way the research is presented is, not any easier for them to follow than it is for some of us community folks to follow. So that makes a difference too. Bill, I'm assuming your hand is still up from before, right? That there's not something new.
Sorry, I didn't know you had to take it down.
That's okay. I just wanted to make sure you weren't wanting to say anything. So again, so this quote, anyway, it's a little bit scary to me, gene therapy. And I think patients also maybe rightly would be a little wary of it, really did echo what we had heard from folks. Oh, I'm sorry. Next slide.
Next slide. Yep.
I moved ahead of you in my own version.
That's fine.
So, but I think that really kind of sums up how people look at it. There's still a lot of discomfort, I think, with this particular aspect of the field. Next slide. One of the things we asked them about was how they would feel about delaying treatment in order for somebody to be in a particular study, if somebody needed to actually be treatment naive in order to participate. And they felt that there was a lot of conflict between best practice in care, which is to start immediately, and delaying care. They were concerned about, well, if I tell people they don't need to start their medicines right now, what does that mean about the importance of actually taking medication? How would it impact the provider-patient relationship? And would people even come back for care if they were involved in a study? Would they think, okay, well, I'm getting all of this stuff done in the study, so why do I need to establish a relationship with this particular provider? How long the delay was made a difference. Some people were okay if you had somebody who had, low viral load, very strong T cells. If you were going to delay them for a few weeks, some people felt like they could delay up to a year. So there were a number of things. Was this a sexually active person? Because we now know, if you're undetectable, that's untransmittable. And what would that delay mean in relationship for that? So they really wanted to know about patient level factors, medication access, their psychosocial state, comorbidities, what is their ability to adhere to partner protection measures, as well as trial-related factors. What's the monitoring plan? What kind of communication plan would there be with the primary care person? So those are all things that were contributing to how they would feel about that. Louis, did you want to say something?
Yeah, I have a clarifying question.
Uh-huh.
So the lack of enthusiasm for cure research that you mentioned earlier that you found in the survey, was that specifically in general in terms of cure research, and it's a far off concept that people don't believe is going to happen anytime soon? Or was that a product of the methods of cure research like gene therapy specifically, and how the quote mentioned, like that's a scary prospect for patients?
I think it had more to do with they see a cure being, a decade or more out. And they have, treatment things that are changing right now that they need to stay on top of. I think it was really more about the imminence of a cure rather than necessarily because it might be gene therapy or not.
Okay.
Yeah. That was my perception of what they said.
It's
So,
still a little disappointing to me that
Mhm.
that's the feeling. Like, I know I can get why they feel that way in terms of actionable things to do for their patients right now. But just to be able to reiterate that hope and that understanding that cure research is something that's happening and could be possible, to me, that seems like it would be important, especially for a community that is chronically dealing with a disease. It would seem like yes, we have effective treatments, but at the same time, having a cure and having to potentially not have to deal with this disease anymore is just, I don't see how that wouldn't be as a big of a development as anything for people living with HIV.
yeah, I think they thought it was important. I mean, they understood that, they clearly understood the psychological importance of a cure. They certainly understood, the impact of stigma. They certainly understood that, there's still ongoing damage to the body, and, that it would be nice to have something that would stop that, right? But they also, in the hierarchy of what they're giving their attention to, they're certainly giving a lot more attention to treatment than they are to cure. And I think the fact that, we haven't had active cure studies happening around here, right? So there hasn't been something, there hasn't been much for them to engage with around cure research either. So I think the combination of all of those things. So yes, I think this would be a good thing. I think it's an important thing. But I don't think it was at the top of their list of what was, most important.
That makes more sense. Thank you.
Yeah. Next slide. So again, these are some of the kinds of things that that the providers said. Again, around, being concerned about their level of immune suppression. How long is the delay? How risky is the intervention? And they're, giving me pause is, a place that it's showing up. And then there were people who said, hey, if somebody was like basically doing well and they were really motivated and it wasn't a high risk study, I would be very supportive of my patients participating in that. Next. And then I think one person particularly emphasized, starting HIV meds is not an emergency, right? Even though right now the current standard of care is, can we get people, same day access, and we know if people start immediately that they're quicker to be suppressed, and there are all kinds of good outcomes from starting people right away. For many, many, many decades, we didn't start people right away. And, people sometimes will want to wait for a genotype test or something else, which, can take a while to come back. So, so all of those things contribute to how people felt about delaying. I would say that there was a lot more concern, it seemed to me, about delay than about interruption. Because delaying is really, that's about getting their relationship with their patient established. It's about, what are the messages you're giving about the importance of treatment. And a few of them actually thought it would be hard to find patients willing to delay treatment because, folks with HIV have heard start treatment soon, right away, the same as the clinicians have been hearing that. So I thought that was an interesting point. Next. So again, I think I need the next slide.
Okay.
So we've had, providers have had issues where when people got into studies initially, they just didn't come back to care, because they felt like they were getting sufficient care from the study site. And so really having some kind of a partnership with the researcher, where the researchers are also emphasizing to the participants the importance of showing up and being at their visits. So monitoring frequency and viremia also affect support for when we asked about ATIs, people had a lot of the same concerns that they did with treatment delay. So really being clear about what kind of monitoring was going to be done. They were worried about establishing resistance if there was viral rebound, being clear on what were the stop and restart criteria. In general, the longer somebody was going to be allowed to be viremic, the more uncomfortable they were with that. Again, they wanted patients to really be clinically and immunologically stable to participate in an ATI. They wanted to make sure people really understood the risks. There was a lot of focus on the importance of informed consent and making sure people really knew what they were consenting to. I would say partner protection was a huge issue. We had people say they thought PrEP should be provided for free to people's partners, and that if people weren't willing to abstain from being sexual, if they didn't have a regular partner, they weren't sure that that was a good person to be having a treatment interruption, having very clear entry and exclusion criteria. They were very concerned about the patient's emotional stability and life stability. What happens when you think you might be cured and then you find out you're not? And we've certainly had Defeat HIV has certainly had people who have had that experience come in and speak before. Again, having good communication plans between the researchers and the providers is also really important. Next, please. So again, this was somebody who felt that, again, it gives them pause, but really worrying about extended viremia and what that might mean. And again, supporting somebody, though, if they really wanted to participate in the research, how do they help support that? Next one. So again, this is somebody who's worried about risks to their partners, as well as potential harm to the patient's physical health. But they also know that often, things with HIV can develop more slowly. So that there might be time for looking at that as long as there was very, very close monitoring. Next slide. So again, for people who are having treatment interruptions, really important for that person to be somebody who could be tracked down. So this person was worried about, are they housed? Do they have a cell phone? Can they come in as often as they need to? But all of those kinds of things were the kind of protections that they would want to make sure were in place before somebody underwent a treatment interruption. Next slide, please. Again, an emphasis on robust informed consent, PrEP for partners, and agreements people make around avoiding sexual transmission during an ATI. That's the treatment, an analytical treatment interruption, in case anybody is unfamiliar with the term ATI. And then next slide. But ultimately, I think this person really said it well, that you have to believe people have the fundamental right to make terrible decisions, and that's, autonomy. And so whether they like the decision or not, as long as people really understand what it is they're consenting to, that they have the right to do that, and that as the person's provider, they would support them doing that. So. And that's for any trial, right? Cure or otherwise, with or without an ATI. That's just a general principle. So next slide. So again, when we asked them about what they perceived to be risks and benefits of participating in cure trials, and they felt that for the most part, a lot of the potential benefits were socio-emotional, feeling like you're contributing to cure discovery, giving life meaning after a diagnosis. There's also the hope of possibly being cured, but again, that carries an emotional risk if you wind up becoming disappointed. Being in a trial could be very stressful. But then there's also the potential benefit of freedom from ART. Not having to take meds on a regular basis could really be liberating from somebody, for somebody.
Mhm.
And we asked them what they thought too much risk might be. And people spoke to excessive toxicity and even risk of death. They also thought once somebody's off meds, it can be harder to get people, back on meds, getting back into that grind of taking daily ART for those who are on it daily. And again, the risk of sexual transmission was a big one for folks. So next. So we asked them about recommendations that they had around a number of different things, like what would they want trial researchers to think about or to do, and these spanned a couple of different areas. Next slide, please. So one was about keeping them informed about HIV cure research, and this was an area of very big interest, I would say. And what they were interested in is, how can I easily find out about open cure trials that my patients are eligible for, right? So not a study somewhere off in Pennsylvania, not a study happening in Thailand, but what's happening right here that people could participate. And so they talked about wanting, one person suggested having a searchable website or an app with all the information on entry criteria and aims and that kind of information. Now I know TAG has a great website that looks at this, but it's got global information. So the idea of having something that would be more localized, study information that would be easy to give to their patients, right? With contact information and that talks about it, or a laminated sheet with information on open trials, having researchers come and talk at one of their meetings about what's happening with the studies, and having an actual phone number of a study staff person who could answer questions if either a patient or provider wanted to learn more about the study. I mean, basically, their message was, make it easy for us to know about and refer to studies. Because it seemed like that's not always information that's easy for them to have access. Next slide. Yep. As far as it advances in HIV cure research, again, they kind of wanted somewhat of a low bar in order to get high level information. They're really not interested, by and large, most of them aren't interested in like a lot of the very nitty gritty information. They really want kind of high level, where's the field, what's happening, what seems to be working, what seems not to be working. So, after a workshop like CROI, having, a session for in a CROI review that has an update on what were the high levels of cure research presented at the conference. Same thing for, IAS. Also, at some more community-oriented conferences, having sessions that focus on cure, providing CME for it, having online seminars, so having recordings of sessions like this that people could access at any time that's convenient for them. A lot of times, providers are busy, they're in clinic, it's hard for them sometimes to get to programs. So having things that they can access at their leisure. The kind of pre-conference that we're doing at CROI, making that available to clinicians as well as to other community members. Next slide. Some other suggestions, I would say they were split on whether or not somebody would be interested in like a simple email update or some kind of periodic email that summarizes this kind of information. Some people said they were drowning in email and were not the least bit interested in seeing another email. So, you know, again, there was a lot of variation in provider preference on that one. Also, if there were pharma-free podcasts or Twitter feeds, that could be another way of updating people on what's happening with the research. Next. So suggestions that they had for trial investigators, again, this online repository, having simple trial referral mechanisms. Next, study recruitment materials in plain language, having phone numbers. So these recommendations, in our paper, they're all summarized in some tables that make them kind of easier to look at.
Yeah. I just made it this larger to read because those were so small.
Right, I know.
Yeah.
So next.
Yep. This and then.
So you know, I think, you know, having some kind of an FAQ, something that might answer all of their questions. I think a written plan for how and when the study labs and other information will be shared with clinicians, because often, you know, there's kind of like a brick wall between a study that's happening and clinical care. So if it's possible, you know, to share those kind of lab results with the clinician, so that the clinician knows, oh, what's being measured and when will I be informed about it? How will I know if there's a change in my patient's clinical status or their immunological status? That those things be communicated and they know what the communication plan is. Next.
This is the part of the chart that was about the suggestions for the community clinicians.
Yeah. So we had originally really started with recommendations primarily tailored for the investigators, because we felt like, you know, the investigators would be highly interested in having the clinicians make referrals to their studies, and that investigators were likely to have a higher level of motivation and doing what they could to make sure that the clinicians were supportive of the research and referring their patients. But it is also incumbent on community clinicians to be aware of, you know, what is happening and how could they get interested patients involved. So certainly there's clinicaltrials.gov and there's TAG's Cure Trial website, so people could avail themselves of that. Finding out the best ways to become familiar with local trials, taking advantage of education opportunities. One of the things that CROI does is they do have a symposium for you know, new investigators, people new to CROI, and even if you're not an investigator, if you are a community clinician, you're going to be attending CROI, that's something that you can avail yourself of, or the pre-conference that we're putting on. So taking advantage of these opportunities that are a bit less sciencey than some of the data that would actually be presented at, say, something like the HIV persistence meeting in Miami, right? That gets way into the weeds in a way that is really beyond what they would be interested in. So, you know, basically, the easier it is for them to refer, and the more they know about what will happen, the more engagement we're likely to see from community clinicians in these things. So, let's see. Next. So here so our key takeaways, just to reiterate, next slide, is that they were generally supportive of participation in cure research and research generally. And they were willing to facilitate referrals as long as it was easy and they were clear on what was going to happen. They do believe that cure is important, but again, it is a lower priority than other things for them right now. Next slide. And researchers can really take advantage of the opportunity to foster relationships and do bridge building in advance of launching trials. Talk with the local providers and see if anything they're doing might feel unacceptable to them. Just like we were talking about the ACTG study, right? Saying, oh, is like 100,000 copies for 3 weeks? Is that an acceptable range of time? Would I refer to somebody to that? Is there a different number that might be feasible? Is there something about the study procedure that seems like, oh, that's a bridge too far? So having those conversations before everything is set in stone may be a good thing to do. And then as those relationships are built, I think it's also more likely that referrals could increase over time. If you want to see the actual study and read all the quotes from all the providers, our next slide has a QR code where you can access the paper. And you don't need a journal subscription in order to access it. It's free, open access. So we've got more detail there. And then next slide is, I just really want to thank everybody who participated, the people who took the time. And I have to say, when I did these interviews, it was at the height of the COVID epidemic. So people were really, they were snowed under. I think it's also part of why we didn't get a more robust response, is people were, they were just drowning in COVID at their clinics. So it really, I think, took something for people to participate. And we were really very appreciative of that, of the CAB, whose idea this was in the first place, of our research team, and certainly, the NIH for the funding. So I'm happy to answer any other questions people have. And if anybody else who was on the study team wants to say anything or chime in, feel free.
Yes, and thank you very much, Laurie, for presenting all this. I know it was a lot to go through. I just wanted to thank you for that. And I don't know if everyone was paying attention to what was going on and being discussed in the chat. But if you had any questions that you wanted to bring up or any thoughts that you had, I know Lewis's thought was shared by me, like what he was saying about it seemed to be a little bit of a lackluster response when clinicians were saying that they weren't interested as much in cure, that they didn't have the brain space for the cure info, that they were focused so much on treatment. I did wonder if that doesn't create some sort of unnecessary barriers. But I don't know how other people felt or thought, or what you think of what this information presented.
I have a thought that my impression is this, that the clinicians themselves are not exactly most enthusiastic as specialists in this field, practically practicing medicine with those who are very involved in this particular area, both research and treatment. I'm rather shocked in a way. And then my impression is that they themselves are not very informed. It is a very small number of sample, and I'm not sure that this is a true representation of the spectrum of clinicians that are sharing this kind of an impression that is given to the audience, like this one here, who is rather involved in the world of HIV research, as well as being community members. So those are the comments that I have to offer for now. I'm quite sure that there are many excuses and many reasons for not having enough clinicians' involvements and interviews and so on and so forth. I do understand that very well. With that predicated to my comments, I realize that perhaps I, as a member of the CAB, should be a little bit more active in order to be able to recruit them and get them more informed and involved.
Well said. Other thoughts?
Yeah, and I you know, and I don't think we can say a sample of 12 clinicians, and as Bill pointed out, primarily white clinicians. You know, we can't say that that's representative, right? Generalizable to all the people who are providing HIV care in the world. But I do think that I think it's significant enough for us to pay attention to. I think that most clinicians would be concerned about the same kinds of things they were concerned about, right? Like, is this person healthy enough to participate in a trial? Is this person, you know, psycho-emotionally and socially stable enough to really be able to participate in this trial in a way that's good for them? So, you know, I felt like they were coming from a place of concern for their patients and what would be best clinically for folks. Very much.
Jared. Yeah, Jared.
Hi, yeah, that was a really great presentation and really important work. I found it really poignant that you asked the question about people's concerns or people's considerations in terms of delaying treatment, because I really feel like this is a trend where the HIV cure field is going in terms of having interventional studies at the time of ART initiation or slightly before. I was wondering if you've got any thoughts about how we as HIV cure researchers can address those concerns before we get to the enrollment stage, which may have some difficulties.
Yeah, I think some of that might be about like what are your inclusion and exclusion criteria. I think you know, who is it, who might it be safer to delay treatment for? But I feel like most importantly, the communication plan with the provider, and how are you going to reinforce, how can you both be reinforcing the message of, even while you're participating in treatment, even if I'm not providing your ARTs right now, how are you establishing care with me? What are we expecting to have happen with the research team versus me as a clinician? What are you as a researcher going to be communicating with me as a clinician? And again, how long is that delay? Because there was definitely a lot of variability in the tolerance for how long a delay would be acceptable to people.
Thanks.
And
well, I have always thought that a cure would not happen in my lifetime. And that's not saying much because I just turned 70. But if you look that I tested positive in 1986, that's quite a stretch. And I've told this story several times that, you know, when I tested positive, I read an article in the New York Times stating that it would take 10 years for them to find a cure for HIV. And here we are, still waiting.
Well, I remember back
very
in
easy to think that it's not going to happen in my lifetime. And I noticed that when the medications really set in and started working, it's like the urgency for cure research kind of fell away.
Yeah, I think too, you know, I remember back, it was either 85 or 86 when Margaret Heckler said we were going to have an HIV vaccine in 2 years. And, you know, here we are 40 years later, and then look at COVID, right? Within 2 years, we had a COVID vaccine. So it's really hard to predict, what's going to happen. One of the people that we interviewed spoke to how seeing the COVID vaccine develop really made her say, oh, well, it might be possible to technically do this, but then feeling very pessimistic about having it be accessible, all the places in the world that would need to be accessible once we discover it. So some real recognition of how social factors and, things play into stuff. And I don't know if everybody noticed, but Michael actually put a direct link to the paper in the chat. So if you don't have the QR code scanner, you could just get the paper there.
Exactly.
Bill?
About freshwater, yes.
Well, I think that's one of the good things about research is, there are also groups that are working on accessibility. So it seems to work, it's not just one person doing everything.
Yeah. Okay, the other Bill?
Hi, a great presentation. It was, for me, very interesting to think about it, recruitment to studies from the point of view of the clinician, because, every time in disclosure, I did an ATI study at the NIH. I think as the ATIs get longer and more involved, and a group of us age out of study participation, the real question to me is, where are we going to find people to be in the study? I mean, it's equally, learning about the study, but where are these people going to come from? The large number of people that we need who are going to be willing to do a long, my ATI was, I think, 72 days. And yes, it was magnificent to not take meds for a while. I mean, just, from somebody who's been positive for as long as I have, and, lovely. But it just seems like they're getting longer and longer. And I worry about where we're going to find the people to do it, but some really great points. Thank you very much for the presentation.
Yeah, related to that, I think the technology we have will also make a difference. When you think about, they have these buttons you could put in your arm for 2 weeks to measure your blood sugar, multiple times a day. If we had something like that could measure your viral load, and you could know in real time if your viral load was coming back, think about the difference that would make in someone's, willingness to have an ATI and a clinician's willingness to support an ATI, right? And, they did speak to, I don't know if you're going to find people who will delay treatment, right? So we do need to look at what is best practice and what are people willing to do. And it really is all altruism. And I think you're right. It is an older cohort of people who are really aware that it was the early people who participated in trials back when we knew nothing, that paved the way to where we are today.
Mm-hmm.
So,
Rachel.
Yeah, I wanted to respond to something that you said, Laurie, about kind of the different responses that people have to how quickly the COVID vaccine came. Because I think, I've heard kind of both this positive, like, oh, if we can do this and we can get an HIV vaccine or an HIV cure, but I've also heard people say, well, clearly this is because we don't care about HIV. And I think that in talking to the people who are working on this as well, HIV vaccines, I feel like I would include myself as part of cure research from the clinical trial standpoint. It's only ever been more immunologically complex than I think anybody would have imagined. And getting such a highly effective COVID vaccine, at least for the first 3 months of efficacy against the ancestral strain, was much more immunologically easy than anybody would have imagined. And so I just wanted to just kind of put that out there, because I've been hearing a lot of sort of misinformation about this is due to lack of care or lack of attention. And I think that, there's still a tremendous amount of effort, both in the labs and clinical trials, in trying to make this happen. And I think that a reinvigorated effort on this from like speaking as a scientist because of some of these things that we've seen, broadly neutralizing antibodies against COVID, vaccines against COVID, and not the opposite. So I guess I just wanted to sort of put out that reassurance that it's not due to lack of care. It's due to, wow, we thought we'd be able to solve this problem, and it's seemingly much more complex.
Right.
Yeah,
there's always multiple sides to everything. I just felt like I had to say that because I talked to a lot of my patients say this to me as well too. And I feel like it hurts in my heart a little bit as someone who's both their doctor and as a researcher, because it's like, no, we care, we do. People are working this very quickly.
Yeah, I think that, I often tell people that the COVID vaccine was built on the 40 years of HIV vaccine research, and it's the HIV vaccine research and infrastructure that allowed the COVID vaccine to be developed as it was. And yeah, I mean, HIV is a retrovirus and COVID is not. And so the differences in that, the challenges, we've never cured a retrovirus, and we, to my knowledge, don't have vaccines against them either. So
yeah, and truthfully, we're not really, like, the vaccine against COVID is protecting people from dying because it's allowing the immune system to get a bump so that it can sort of get a head start on it. But the current efficacy of a vaccine against a new infection is, like, by best estimates against XBB, this new variant that's out, is like 17 to 40%, right? So that's in the range of like the Tyvek, the RV144 vaccine, maybe. So I don't know. Anyways,
I think it's how we get
and the new HIV mRNA vaccine has sort of catapulted on the COVID vaccine, which, as Jared put in the chat also, was accelerated by the technology from the HIV vaccines too. So
go ahead, Bill.
Well, I think the average person living with HIV doesn't know the difference between a retrovirus and oh, that's why.
Right.
And so I think as we go along, and here we are at what, how many years, all these questions that keep coming up, and there's still this group think that, it's a government.
Yeah.
I hear
and
that more.
So much in the news where people are talking about, like, the immune system is depleted with COVID, just like with HIV, and it does the same thing. Like, there's all of these crazy scare misinformation things about COVID, sort of parlaying on things about the immune system and stigma about HIV. And so that, I think, it only makes it worse because almost all of that is immunologically untrue.
Yeah. Yeah, like the messaging is challenging, to make it simple enough and accessible enough and clear enough for people to really get it. And then, of course, it's what do people believe? Because we all bring our biases to everything. And now that all the information we get is known to be slanted in one direction or another, it's really hard for people to have confidence in any particular source.
Yeah, I think
I do see
no, I was just going to say, I was a part of a webinar 2 weeks ago, and this gene therapy researcher said it in a very simple way, that HIV has been a far sneakier opponent than anyone ever thought. You know what I mean? So when we heard back in the 80s that we'd have a vaccine in 2 years, that was based on what they knew about viruses at that time. And we've learned so much since then because of HIV, because HIV is a far sneakier opponent. I mean, just the way it infects part of our immune system and destroys it that way, that, our own protections are not catching up, and how it makes millions of copies of itself, each of them a little bit genetically different, which become like trash that serve as detritus that kind of mislead us, and take our immune system a long time to catch up. I think all these things that we've learned from HIV research really is just proving, but we're knowledgeable about that. The average person doesn't know anything about that, but they know about what it feels like to have an uncaring government who doesn't care that you're dying. And I think that's the problem that we have, is that the lived experience of people doesn't match with what we're saying. And so it's like we're trying to play catch up, but I feel like we've been trying to play catch up for 40 years.
Yeah. And I did see a comment in the chat about, what's the difference between a long-acting injectable and, remission. And I think that's kind of where we're looking at, what is durable suppression. If we get to a point where instead of needing an injection every month or two, you need it once or twice a year, I wouldn't call that a cure, but, how does that impact the perceived urgency for a cure if you have access to that, and how different is it if you can't transmit? And a question is there still ongoing inflammation? And I think there's a lot that we still don't know about the long actings. But when we did our research with folks with HIV earlier on, that Scared and Brave study, folks, really, some people thought that the term functional cure, it gets thrown around a lot, and people felt functionally cured if they were undetectable on their meds. They didn't see it as, some folks didn't see it as that different. Part of why I don't like the term functionally cure, because it means so many different things to different people. But yeah, so the lines do get blurred a little bit. But if we're talking about an eliminating cure where there actually is no HIV in the body, which is the cures that we've had, right? Timothy Ray Brown and,
Yeah, the bone marrow stem cell transplants. Yeah. Well, thank you, Laurie, again.
I also hate that term functional cure.
I've been arguing against it for a while.
Well, I just wanted to, we're over time, and so I wanted to make sure that we honor people's time and let people go. But I wanted to thank you again, Laurie, for sharing this with us all. I hope you all found this edifying and you've learned something or gathered something from this. It sure shows that we have a lot of work that we can still be doing in terms of advocates, in terms of new strategies around stakeholder and full stakeholder engagement. There's a whole group of people that might not be addressed that have needs besides the people living with HIV, and that might be the people who are caring for them. And we should be thinking about them. So thank you for this, Laurie, for sharing that with us.
Oh, my pleasure.
For taking the time to join us. And we'll be meeting again next month for February, which will be post-CROI. And so I will be giving us a roundup of CROI research that has been presented at the conference as it happens this year. So you will want to tune in on that. That'll be a community update from post-CROI. So good to see you all tonight, and I look forward to seeing you again in February. Thank you all for joining. Take care.
Bye.
Bye-bye.